Okay, folks, we're going to go over Module 1, Lesson 1.1, talking about the microscope and the cell theory and how it played a role in biology and the future of biology, and we're still using these great advances today. So there's your objectives for the module, Lesson 1.1. Explain how advances in technology have led to discovery and further study of cells. Need it more than ever now with this coronavirus. Identify the parts and demonstrate the correct usage of a compound microscope. Calculate total magnification. Explain how advancements in technology contribute to the cell theory, and describe the cell theory and how scientists, and know the scientists who were responsible for it. So on the bottom here, you see a bunch of just various kinds of cells. Okay, so here's some history. This gentleman here, Anton von Leeuwenhoek, discovered the microscope. He was actually a toy maker and was explaining or playing around with lenses and came up with the first microscope. And you can see the years 1600 to 1700 something when he lived. So he is considered the father of microbiology when he helped develop the microscope. The next gentleman played a role in cell theory here was Robert Hooke. He looked up and was able to see cork cells underneath a microscope. And if you look at the picture on the right, what you see is a cross-section of a cork, and all the little boxes or little channels inside, he called them cells, all right? And that's what monks lived in a monastery, and that's what he attributed similarities to. So the next guy, these are a couple of Germans here, and they're going to help contribute to the cell theory. This particular gentleman was able to recognize that plant cells, or plants, have a bunch of individual units that make up the big plant. His name is Matthias Schleiden, and the next gentleman basically said the same thing, that all animals are made up of these individual cells. And so I always remember the difference. Schwann has the letter A in it for animals. So Schleiden was the plant guy, Schwann was the animal guy. So then this gentleman, Rudolf Virchow, was able to take both of their information, Schleiden and Schwann, plus Hooke, and figure out that the cell theory. And the cell theory basically states three things. One, cells are the basic unit of life. You can't get anything smaller. Cells come only from other cells. They don't just appear out of nowhere. And all living things are made up of cells. So that was Rudolf Virchow came up with the cell theory. Okay, so the microscope, basically you have lenses which are able to bend the light and increase the magnification, or increase the ability to see small things. And so we'll look at how we utilize the microscope, and there's an activity in the learning menu on how to use it. So pretty simple. You have to have light be able to come through in order to see your subject. These two particular microscopes are vastly different and do different things. The one on the left, very affordable. You can, that's what we basically use in the classroom. The one on the right, very expensive, but you can see way more detail and smaller things. Just an opportunity here to kind of get some comparisons on the various size of things, down from a nanometer or parts of a nanometer all the way up to 10 meters. Resolution and contrast are two major items to talk about when we discuss the microscope. Resolution is the ability to see real detail, and contrast is help with light. When you look at a microscope, when you're able to see the space in between two lines, that's resolution. And the better the resolution, obviously, the more detail you get to see. Okay, here's a picture of a plant cell called Elodea, and there's a little chloroplast, or the green little organelles inside of those cells. But the compound microscope, again, uses light to help illuminate your subject, your specimen. A couple of lenses, and then we'll look at how those lenses help magnify here in determining magnification. So here's your typical microscope. All right, so we have the lens up here. All right, so where we put our eye, and it's usually around 10 times. All right, so it's helped magnify something 10 times. The light will pass through here, and this is your objective lenses, and you can manipulate those. Generally, you have a lens that is a 4, a 10, and a 40. So if you multiply the top here by the 4, it would be 40 magnification. So you would be able to see something that's 40 times larger. If it was a 10, 10 times 10 is 100. And then if it was a 40, 10 times this lens times the 40 magnification or objective lens, that will give you the opportunity to see something 400 times larger than what you normally see it as. Okay, so then the other things are just parts of the microscope to help you locate and identify and find your objects. All right, so here's your arm. This is what you hold it with. The fine focusing mechanism and the large coarse focusing mechanism. This one up top here, the coarse, you generally start with, and then as you get into more detail, and you don't want this stage to move very far, that's when we go into the fine mechanism. So generally, we're using the coarse with the lower objectives and the fine with the higher objective lenses. Okay, and then just the other body parts, parts of the microscope here. The stage is what you put your slide on. It goes up and down. There's some clips generally up here to help hold your slide down so it doesn't slide around. Most of our microscopes nowadays do not have a mirror, but they have an electrical light that shines light through here so you can see it. Okay, again, here's how you can find your magnification. We just went over that. Just multiply the eyepiece by the objective lens. In the Hilliard City Schools, most of our microscopes have the maximum magnification of around 400. A few of them have ways to make that higher using oil and some other things. Right, here's a way that we can add or subtract light, and that helps with contrast. It's called a diaphragm. And so move it one way, it gets darker, or it allows more light in, or it takes away light. Okay, go look through this. Pretty simple. Start on the lowest objective lens and just pull the slide up or the stage up towards your eye. And as it comes into focus, you use the coarse adjustment. Look through that, make sure you're aware of that, and it will help you with your activity in the learning menu. All right, so you look here in the light microscope versus the electron microscope, big difference in the, on the left-hand side, you can see what they're talking about, and then you compare the two different microscopes. So in order to see something on the light microscope, you need lights. In order to see something with the electron microscope, you need electron beams, right? So there's a big difference. Magnification, you can get up to 2,000 with the light microscope, and here up to 500,000 in some of the ones today, or even greater than that. Resolving power, 200 nanometers, right? So the size of, being able to see something, whereas in the light microscope, it can be significantly smaller, only 0.2 nanometers. Excuse me. How do we focus? With the light microscope, we're using lenses. Over here with the electron microscope, obviously we're using electromagnetic magnets. Okay, here's the big difference between these two as well. You can see something that's alive under the light microscope. In order to prepare the specimen for the electron microscope, you have to kill it. So it's going to be a dead item, so you won't be able to see breathing or moving parts or what have you. Light microscope's very small, and the electron microscope's enormous. You can see materials to make these things very simple to complex and cheap versus expensive. And that's it. Let me know if you have any questions. Continue on now to the learning menu.